Good morning and welcome to our conversation today that picks up where we left off last week in our newsletter when we were talking about repurposed drugs and how some of these drugs use some very interesting biochemistry to tackle disease X. And one of the approaches for these drugs pharmacologically was to work with microtubules. And I didn't at that point talk about what microtubules were. I was just listing the different areas that the repurposed, or even new drugs for that matter, development of new drugs, using microtubule biochemistry to impact cellular replication and various other things. So I thought, okay, this is really too interesting to pass up. So I thought I would put together a little lesson, so to speak, a little conversation about microtubules in every single one of our cells and how awesome they are. So today we're going to define what microtubules are and how they're constructed, which is interesting. We'll show how microtubule influence cell division, which of course is related to all kinds of medical applications and disease applications and wellness applications. We'll identify some very interesting structures that attach to the microtubule. We're going to do a little abbreviation there, and how those attachments, those little structures on these microtubules, identify the particular microtubule and cell type, and how microtubules have been targeted for drug development. So let's first look at what a microtubule is, and you're going to just latch onto this and say, wow, this is really crazy good. And so I have a wonderful diagram here of a microtubule. The interesting part about this is what you're looking at is very, very tiny. It's hollow. These little areas inside this spiral is hollow like a slinky, and the structure is made out of something called tubulin. Tubulin is a protein type, and we have two types. We have alpha tubulin and a beta tubulin, and they're assembled in this spiral way so that one side of the tube is going to have a positive side and one side is going to have a negative side. That's really interesting and very helpful from a chemistry point of view when structures can align themselves up like a magnet. So these microtubules are then assembled into larger structures. And so let me show you this wonderful micrograph, an electron micrograph picture of two tubulin structures. Now these are like cross sections, and I don't know if on the film it really shows up, but you can see individual microtubules are all assembled in these larger tubes. So each one of these has many, many, many individual microtubules making up the structure. And so you can see that the arrangement of these individual microtubules dictate the type of larger structure that can be assembled. And when you're talking about biochemistry and putting different proteins together, the combination is almost endless, which in and of itself like boggles my mind. And then over here is just the simple picture again of the alignment of the alpha and the beta tubulins to make a particular microtubule. Well, what makes these so special? So let's just do a quick little biology class about simple cell division. This is just going to take one moment. We have a simple process that you might recognize where we start with the cell starting to replicate by reproducing the chromosomes, okay? And so, but what's so crazy is out from this center port called a centrosome, microtubules are produced, and they attach to various chromosome locations of the replicated strand. And then what's so great is that because these microtubules are dynamic, meaning they can change shape, they can change length, they can change all kinds of ways, the microtubules start to shrink. And when they start to shrink, look, this size is much smaller than over in the newly established dividing cell. And when these microtubules start to shrink, guess what? They start to pull apart the replicated chromosomes. So the dynamic nature of these microtubules totally control cell division. And then finally, as the cell starts to pinch off, you get a reassembling of your chromosomes into the nucleus, and the cells can separate, and you get your two new cells. Without microtubules formation, way over in the beginning, if you don't have a good, steady, healthy, accurate microtubule production, positive cell division cannot happen. On the other hand, though, if you have cell division that you don't want, i.e. tumor, metastases, and growth of different tumors, and you have uncontrolled cell division, maybe controlling your microtubule is the answer. And that, in fact, is the field of drug development right now for controlling all kinds of growth processes in your body. So let's see what the drugs are targeting. Here are a bunch of microtubules, and you have individual microtubules that are connected to each other, making the larger ones, correct? And in between the microtubules, you sometimes get these other structures. You get other receptors, maybe. You get other things called microtubule associated proteins. The maps are very specific to different microtubules. So attacking and understanding maps in cell division and cell production and cell health, understanding the maps may be a ticket to target drug development. And that is exactly what the drug that we were talking about last week, that I didn't name, but it was just the idea of it, was getting repurposed, and it was proven that that particular drug attacks MAPS and causes upset in the cell division of some cancer cell lines and other whatever cells have that particular map. So what's interesting is the natural world. You know I was going to get to this. Our natural plant world make compounds that support optimum microtubule production and action within the cell structure. And so our sulforaphane, for instance, and you know we've talked about sulforaphane a lot, that's the metabolite that comes from the brassica plants. And in your body, sulforaphane does a host of biochemical reactions. And it turns out that sulforaphane also disrupt microtubule production. And so we have research now coming out of various institutions that are starting to study the link between sulforaphane and other plant metabolites with commonly used chemo drugs. In this case, we're using a very, very standard chemo drug to talk about when you add sulforaphane with the drug, you get a reduction in resistance to the chemo drug. That's really, really, really important. So many chemo treatments and other treatments for disease, we get resistance. And so if we can add something to limit the resistance, we're ahead of the game. And this particular research demonstrated that microtubule disruption was the cause of the reduction in cell division and the association between the sulforaphane and the chemo drug posed not any additional side effects. And so here at the end of the abstract, it says, these findings will help us develop low resistance and high efficiency chemotherapy by metabolite combination. And I think when we think about nutrition and we think about our food and we think about all the phytochemicals, that's what we're after. We're after a combination that's going to prove to be most efficacious towards wellness. So I hope that the discussion of these microtubules in a nutshell and how they start off as proteins, they assemble themselves, the microtubules themselves assemble even to higher order structures like we saw in the micrographs, and how the dynamicness of these microtubules to expand and contract support cell division, and therefore we can use those microtubules as a target for the development of drugs to control diseases that are about rapid cell division. So next week, or the next YouTube that I make, is going to be about another area that these repurposed drugs can control, and that's using something called a tumor suppressor gene, a p53 gene, and I hope to have that for you next week. Have a wonderful day.